Good afternoon. Thank you very much for coming this afternoon. This is an open-ended discussion, not open-ended for the time, open-ended discussion about what has come upon us as the H1N1 flu. So we have three panelists. Myself is Adel, Mahmoud, and next to me here is Lynn, Enquist, and Kahn. And we'll try in the first 25-30 minutes to cover some of the basic information about virology, about the virus, about our relationship with microbes, and about our responses. And the idea here is, through the three opening presentations, is just to cover some of the basics and some of the general directions, and then leave it up to your questions and answers where we would want to engage into a conversation and a discussion. So that is the plan, and I will start by just explaining a little bit of the relationship between microbes and humans. And we like to call this the convergence model, which came out of a study about emergence and re-emergence of microbes as agents of disease. In the center is us and the microbes. And the conceptual framework is to appreciate that we both are co-inhabitants of the globe. And as a matter of fact, there are more of them than of us. So consequently, it is very, very important to appreciate the complexity of that relationship. We have existed since the development of this globe and its inhabitants, and we probably will exist as long as the globe exists. And consequently, it is very, very important to understand why this phenomenon, which we call emergence, every now and then come to challenge us. And the feeling of the scientific community, and this is coming out of the intricacy of the interaction between us and the nature around us. So, for example, changes in our genetics and our population factors, the biological factors, are an important element in why some of these microbes sometimes cause disease. For example, there are a considerable proportion of the global population today that are immunocompromised. Their immune response is lowered because of age, because of disease, because of other nutritional factors. The consequences of that, some of these microbes that are under reasonable conditions do not attack us, do not cause us disease, become disease agents. Of course, HIV is the most famous of these organisms, but SARS is another one, West Nile fever. There are several agents that didn't come until there has been something that happened in the human population. On the other hand, the social and political and economic factors, the unrest that is going on, the conflicts that are going on in many countries in Africa, for example, in Southeast Asia, are responsible for peoples migrating from one area where they know the pathogens that are in that area to areas where they don't know the new pathogens, consequently they appear. Ecological factors, whether you believe or subscribe to global warming or not, there is no question the spread of some of these infections is because of some of the warming effect that's going on, some of the loss of vegetations that are going on, some of the changes in the ecological systems, whether there are new dams or new water irrigation schemes that really result into the emergence of these infections. So that is the general field, which means we as the human population on this globe should expect that microorganisms are going to come out every now and then and attack us. Now, the H1N1 is a very, very special case, but it really is something that we have known that it will probably happen. So from the year 1500 onward, there has been several pandemics of flu, of the influenza virus, that came and attacked the population in different forms. The most famous is the 1918, which basically killed approximately 50 million people in this country. But there has been some pandemics before, and there has been some pandemics after. Now, is this current H1N1 a new pandemic or not? We have to define what we mean by pandemics, and my two colleagues and I will try to go into these definitions. But remember, in some ways, the transmission between humans is an important factor in determining that. The coverage of more than one country and more than geographic continent is another factor in that. And then the disease burden that occurs because of this infection is the third factor that determines whether we have a pandemic or not. So these are the three issues that we have to deal with as we discuss H1N1. In 2005, I had the pleasure of chairing a meeting at the Institute of Medicine about the threat of pandemic influenza. And at that time, we were discussing theory. We were not discussing something upon us. And we posed the question, are we ready? And the answer was no. So basically, in 2005, this globe was not ready to face a pandemic of flu. The consequences of what happened, starting with the reporting of the H1N1 cases, has to be appreciated within the context of the framework of, in 2005, we were not ready, and we want to understand the sequence of events that happened since the report of the first case. And Laura will cover that as we go on. So, without spending any more time at this point, what I really would like to do is to introduce Professor Enquist, who is the chairman, professor and chair of the Department of Molecular Biology, who is a virologist. Every cell in his body knows about viruses, talks about viruses, and appreciates viruses. So, Lynn.
Thank you, Adel. That's true. I love to talk about viruses. And so in the time that's allotted to me, I'm going to try to do a couple of things. The first thing is to give you some general appreciations for this life form we call a virus. They're very special, and unless you spend some time to look at them, you don't really have an opportunity to think about how special they are. So I'm going to try to cover 35 lectures of my course in 10 minutes, so we'll do that. The second thing I want to talk about is this special case of influenza viruses. These are very interesting viruses from a molecular and cell biology point of view, but they're also viruses, of course, that we deal with on a regular basis every year, and the H1N1 virus we're looking at right now reminds us of the importance of these guys. But first, just a little commercial. I don't like to talk about viruses as bad guys. They have a phenomenal impact on biological discovery. If we didn't study viruses, we wouldn't know a lot of things about basic biology. There's more than 20 Nobel Prizes that have been awarded for the study of viruses. We wouldn't have genetic engineering, molecular biology, an understanding of vaccines, gene therapy. They all come from the study of viruses. And so many of us study viruses because they can teach us something other than how to deal with disease and bad things. But on the other hand, by knowing how they work and what they do, we really do get a handle on how they do their job. So the first thing I want to talk about is essentially that we live in a sea of viruses. We eat, drink, breathe them regularly. I encourage all of you during this talk to not touch your eyes, your mouth with your hands. And let's see how long you can do that. Basically, we breathe 6 liters of air a minute, and I'm going to show you that every liter, every milliliter of air that we breathe in has virus particles. We eat thousands of grams of food a day, depending on each of us. We touch everything that's possible. We put our fingers in our eyes and mouths. You can't stop from doing that. The concentration of viruses, the ecology of viruses is so poorly understood that only now we're beginning to appreciate the magnitude of their presence on the planet. We know that if you can go out to any place where there's water, this has been done predominantly for seawater, but freshwater is the same. You will find on the order between 1 million to 100 million virus particles per ml of all water on the planet. And we mostly don't know what any of these particles are. And of course, 10% of your own genome is composed of viral, bits and pieces of viral genetic information. And we know that now because we've sequenced the genomes, and we know that viruses are there as well. Viruses infect every living thing. There is not a living thing on the planet that doesn't have a virus compartment to its existence. Our domestic animals, our pets, wildlife, fish, plants, insects. It'd be okay if viruses stuck to the organism that they infect, but many, many viruses, in fact, can cross species barriers. And we have some really classic examples. And those viruses that are establishing a natural relationship in an animal and then infect humans, we call those zoonotic infections. And the infections that Adel was just talking to you about emerging infections, typically the place that the viruses emerge from are animals or out in the wild, and they come into us when we get in contact. From an ecological or evolutionary point of view, this reminds us that essentially these tiny little things are always probing for new hosts, new directions, new places to go. And so where they are today doesn't mean where they're going to be tomorrow. And that's true for just about every one. We know that viral infections influence host evolution, and we know that host responses influence viral evolution. Classic example is smallpox. When European explorers came to this part of the world, they brought smallpox with them. In about 3 to 5 years, they essentially decimated, if not more, the population of the natives that were here. They changed the entire population of the New World. That was smallpox. In 1979, because of the fact that humans were able to think of a way to immunize against smallpox, we essentially destroyed smallpox, natural smallpox from the planet. 1979 was the last case. Other than some stocks that are around, there is no natural smallpox anymore. So you can change things around, and viruses can change things around as well. But the amazing thing is, despite the fact that you're living and breathing and exposed to billions of virus particles all the time, most of them don't do anything to you. The reason why is because we have a defense system. Each cell has a defense system against invading viruses. Each organisms of multiple cells have defense systems. One of the most potent ones is our immune system. We know that this is true because when the immune system is broken, when you have AIDS, you get chemotherapy that takes out your immune system so you can have an organ transplant. Even the most common viral infections can be lethal. So a little bit about what is a virus. These things really require special care to think about what we're talking about. Viruses, sometimes I get questions, are viruses alive or are they dead? Well, basically, it depends on what you're talking about here. The virus particle, the particle that contains the genetic information, is dead as a doornail. It has absolutely nothing to commend it except the thermodynamic behavior of interaction of cells and viruses. So they're not alive in the standard sense. But as soon as that inanimate particle delivers its genome to a cell, then that infected cell takes on a whole set of new characteristics, and those characteristics define the nature of the virus. Now, viruses are some scientists who like to say that viruses are on the edge of life. What's up on that slide is a ruler that ranges from, on the left end, 1 centimeter, about half an inch, down to essentially 8 orders of magnitude to the size of atoms. And where you can see the red arrow is, that's basically the borderline between things that are alive and things that are dead, things that are capable of self-replication and things that can't. And viruses fall just on that line or a little bit lower than that. And the size factor there that we're talking about is on the order of about 10 to 100 nanometers, a billionth of a meter. So these very, very tiny things are essentially complexes of proteins, nucleic acid. Some of them have a membrane that surrounds them. But one thing that defines all viruses is that without a living cell, they go nowhere. They have to infect a cell to produce more particles. So the particle is dead, but the infected cell is something very different. And on one side of the slide are a variety of different particles, bacterial viruses. The long cylindrical one is tobacco mosaic virus. For those of you who smoke, God forbid, you're inhaling lots of those particles regularly. The virus on the bottom, the bullet-shaped one, is rabies virus. And the round one is a virus that causes cruise ship diarrhea. It's a norovirus. So every virus has a characteristic particle to carry its genetic information. We call those virions, and this remarkable molecular architecture is really fun to study, and we know a lot about the molecular detail. Every one of those particles carries the viral genetic information. That genetic information is usually DNA or RNA, and the size of these genes, these genomes, is really remarkable. You have in your genome about 20, 30,000 genes, depending on how you want to count them. Typical viruses, most viruses have less than 15. There's a whole class of them that have only one. There's a bunch of them that have 100 or so, but they don't have very many genes. And so this is very, very important because these are intracellular parasites. They do what they do because they can take over the cell and do their own thing. This idea of being very small is hard for people who don't study small things to appreciate. Something that's 20 to 30 nanometers is really, really tiny. The size of a cell in your body right now is about 10,000 to 3,000 nanometers. So they are 100 times smaller than the size of a cell. Now, the power of things that are small is kind of hard to believe, but the slide here shows a couple of people sneezing. Sneezing is a very common reflex to get things out of your respiratory tract, and when you do so, you spray out an aerosol of the moisture that's inside your lungs. And this is a very common way for respiratory viruses to spread. That is, viruses that grow in your upper respiratory tract and in your lungs can be spread to other hosts essentially by riding the aerosol that your brain produces when it causes you to sneeze. The sneeze range for infection is thought to be somewhere on the order of about 3 feet. The big stuff falls on your shoes and doesn't do anything, and the small stuff goes out and gets in contact with living cells. If these viruses don't come in contact with living cells, they're toast. They have to land on something. And when they're outside the body, depending on where they land, they can last for a matter of minutes to several hours, depending on what surface that they land on. But if they don't land on anything alive, remember that particle is dead. So the power of small is that if you're about 50 nanometers in diameter, and that's the size of about a flu virus, it takes about 100 to 500 million of those particles to occupy a pinpoint, a pencil point, the head of a pin, the size of a droplet in one of those aerosols. So when you sneeze, if you're infected, you produce really quite a good load of aerosol-driven particles that can infect somebody who's within 3 feet of your radius. It's a very efficient way for viruses to spread. Now, this is just sort of a plea to the fact that you can't think about these guys as having any kind of rationale for doing what they do. It doesn't mean that they're simple or primitive, on the other hand. I argue that they're the highest evolved life form on the planet because they survive continuously. Because they need living cells to produce more of them, if they can't get into a living cell, they're gone. The particle is dead, and so they have to survive. But you have to be careful about anthropomorphic analysis. Viruses don't think. They have a way to know what they're doing. They don't plan for anything. The only thing that they can do is provide the material for them to survive. So this idea of thinking in human-centered manner about virus evolution just doesn't work. You have to think about them in a very different way. So here's just one really short summary of why they can survive so well, why they are incredible survivors. They survive because they produce huge numbers of progeny. A single infected cell that's infected with poliovirus produces about 10,000 virus particles. And those particles consist of a whole bunch of mutants. Every time viruses replicate, they make mistakes in their replication, so there are mutations, there's recombination between genomes, as I'll discuss, there's reassortment between genomes, and it produces an enormous amount of diversity. That diversity is selected for in evolution. You have to have diversity in these very large populations because every time that there's a selection, somebody's treated with a drug, someone has a very good immune system, virus particles land on people of different ages or whatever, the particles have to survive and replicate. So the selections are host defense, physical environment, drugs, vaccines, but every time you put a selection on a successful virus population, somebody's going to crawl out. And that's why they're always there. You can't get rid of them because there always is a mutant that can come around. And if you can fix it so that they can't replicate, you can actually stop them from spreading. That's why antiviral drugs and vaccines are so good, because it stops them from replicating, making the diversity that enables them to move on. But it also reminds you that if you don't treat them hard enough, they come back with a vengeance, and what they come back with are things that are resistant that you just threw at them that didn't exactly stop them from replicating. You also have to understand the process of serial infections if you're going to really appreciate the things that Laura and I are going to be talking about in a little bit. The way for a virus to spread from me to you is that, and maintain itself in the population, not just spread to one individual, but to maintain itself, it's got to spread to more than one individual at a time. And ecologists and virologists have a number we use. Laura will also remind you of this. It's called the reproductive number, R0 or R0. It's the number of individuals that are infected from a single infected individual. If R0 is less than 1, you're not going to have much of a spreading virus at all. This is not going to be a successful spreading virus. It'll burn itself out. The R0 for chickenpox and measles, some of our most contagious viruses on the planet, is about 10, sometimes a little larger than that. The R0 for the standard flu that we get every year is about 1.5, and we're calculating the R0 for H1N1 flu right at the moment. The other thing you have to remember is that those viruses that can infect a wide variety of species have an incredible evolutionary advantage. They evolve in one host, can move into another host and evolve there, go back. They can go back and forth, and there's a whole variety of viruses, and influenza viruses are one of them. The one example I'm showing you here are encephalitis viruses that live in birds, basically. They're transmitted by mosquitoes between wild birds, transmitted to domestic birds as well. And occasionally, one of those mosquitoes will bite a horse, and you'll get encephalitis in a horse, or they'll bite a human, and you get encephalitis. But those viruses don't spread from that horse or that human. They're called dead-end hosts. The host may actually die, but the host has no role in the evolution of that virus. The evolution of that virus is in the birds and in the mosquitoes. So viruses that do that are really important. And I think the other thing here is just to remember the evolutionary strategy that's underlying all of this stuff in virology can lead to some very unexpected phenomena. So for example, highly virulent, deadly viruses are the ones that capture the public's attention, that get CNN and the New York Times and everybody talking about them. But they represent an extreme and unstable example of virus evolution. It's going to happen. This is part of the way the process works, but that's not a strategy that really can be sustained for very long, because if you kill all your hosts, you're dead. You can't go anymore. Of course, once you've done it, it's irreversible, and so it's not necessarily a strategy that you would say is high on the table, but it's there. Alternatively, if you are too wimpy to essentially spread around and whatever, you're going to lose too. So the virus population faces elimination under those circumstances. So a big discussion in virus ecology and global health and population biology right now is, why do nasty viruses exist? Is ability to cause disease and kill a few hosts and yet spread very well, is it a successful strategy? Well, the answer to that is obviously yes, this works really well. But it's a big debate, and every time we get a chance to study viruses that are moving around like H1N1, we get to see how good our theories are and how good they match up with reality. So H1N1 2009 influenza A virus is evolution in progress. We're watching it right now in real time, although CNN doesn't seem to be thinking it's as exciting as it was last week. But I'm telling you, it is very exciting, it's very important, and we really do have to watch what's going on. So let me talk about influenza viruses for just a little bit so that you're up to speed on what goes on. There are three types of influenza virus: influenza A, B, and C. A and B are the human pathogens. C doesn't really infect humans. They're distinguishable at the level of sequence. They're distinguishable by the antibodies that they induce. And so we know a lot about these viruses. The first recorded flu epidemic, as Adel told you, was in 1510 in Britain. And every year we basically deal with these viruses. They move along. The virus particles, as you can see there, are they're membrane-covered particles. They have on their surface a series of proteins, membrane proteins. The red arrow points to the hemagglutinin, or the H protein. This is the H of H1N1. This is the protein that attaches the virus to cells. There are about 100 to 200 of these proteins on the surface of a virus particle. On the other side, the green arrow shows you the neuraminidase protein, or the N protein of the H1N1 variety. The neuraminidase protein is a special enzyme that actually cleaves the virus particle away from the surface that it's attached to. The drugs Tamiflu, Relenza inhibit neuraminidase. So if you take those drugs, you basically reduce the virus's ability to spread. There's another little protein that you can see on one side of the slide. It's a little green channel called the M2 ion channel. This pumps protons into the virus particle so that the virus particle can come apart. That's the target for the anti-flu drug, amantadine, rimantadine, Symmetril. That's the same kind of activity. That's an ion channel blocker that affects that one. So basically, this virus essentially has 11 proteins, encodes 11 proteins. It's not very big genetic repertoire. And if you look inside that particle, you'll see 8 little squiggly segments, and those are the genome. And the interesting thing is this nucleic acid is RNA, and it comes in 8 pieces, and that's going to be very important. This is just a list of some information about the viruses. Type A is the virus we really spend a lot of time thinking about because this is the one that really causes a lot of trouble. It infects a whole variety of animals, people, birds, pigs, horses, seals, whales, ferrets, you really name it, this virus is out there. And in that species, each species it evolves slightly differently. There's 16 different H proteins. You go back over here, the red arrow, those 16 different types of that protein, and there's 9 different types of the neuraminidase. These 2 proteins turn out to be the signature proteins that dictate what the immune response is going to be. We know that if you have good antibodies to the H and the N protein, you're probably going to be protected against that virus. And so there's a lot of different combinations of H and N proteins, and these combinations show up in the organisms that are being infected by these viruses. So right now in the United States, the human population here is circulating H1N1, H1N2, H3N2. They're the most common type A viruses that are floating around right now. But all the other subtypes are in different animal species. So if you looked in horses, you'd likely to find H7N7 and H3N8 circulating around right now. We also have a major avian pandemic going on right now. It's called H5N1. And before H1N1, this was the one we were all worried about because this thing has got the capacity to jump from birds directly into humans. But it doesn't seem to spread very well. So we're watching that one very carefully. The type B flu causes nasty infections, but there's no H or N subtypes to this thing. There's only one type. And as I said, type C doesn't really cause epidemics or pandemics in humans. I have a couple of pictures of an electron micrograph of flu particles as they're budding from a cell. In the bottom part of the picture is an infected cell. You can see the membrane as this particle pushes through the membrane and it picks up the cellular membrane that has all of the viral H and N and M2 proteins inside it. If you cut those particles in half, or right down the middle rather, you can see the 8 segments, how they're arranged so nicely, 7 of them around a central 8 segment, so that every particle gets the right number of segments. But those segments turn out to provide flu with an incredibly powerful evolutionary engine. And that engine drives the process of reassortment. What I've got in this slide here is an example of an avian virus with 8 red segments, and a virus that has been circulating in pigs, which has got 8 blue segments. And when those 2 different viruses infect a single cell, you can get all of the segments replicating, and the virus particles that come out are going to have mixtures of red and blue segments. And so I'm only showing you here just the basic outcome, but you can get, of course, the avian virus back, the pig virus back, or hybrid viruses back. The hybrid virus I've illustrated here has 1 avian segment and 7 pig segments in it. But you can get all possible combinations. And who knows what those viruses are going to do when they infect the next round of hosts. And in fact, it is this process that generates the possibility of these viruses to cause pandemics, that is, to infect everybody on the planet. And the reason is because the process of reassortment is generated in some special circumstances. For example, the HN combinations that are circulating in birds and pigs and humans you discover that avian viruses don't infect humans very well, human viruses don't infect birds very well, but pigs seem to be infected by both viruses reasonably well. They have receptors for both. And so pigs are sometimes called the mixing bowl or the reassortment chamber but they're basically where a lot of new reassortments occur. And I'm sure there are other animals other than pigs that have this capacity to replicate both viruses. So what comes out of a pig that was infected with a bird virus and a human virus may be a virus that hasn't been seen by humans for several generations. And if that's the case, there is no immunity. And if there is no immunity, then you have a high probability of being infected and spreading. And that's what we call a pandemic. And I'm showing you in the bottom here a bird virus and a human virus that both of them grow in pigs. And then the pigs infect other pigs. You get reassortments. And what comes out is this unknown quality. The past pandemics that we've had are fairly well established now. There's something called the Asiatic flu in the late 1800s, was possibly H2N2. That time we didn't have sequencing and antibodies, and there's a little bit of information about that. Killed about 1 million people. The 1918 flu was an H1N1 variant that did about 40 million people in the United States. 1957 we had the Asian flu. That was H2N2, a million or so people. 1968-69 was the Hong Kong flu. I had the joy of having that thing. That was not a treat to have, but 1 million people died of that. That was H3N2. 1978, Jerry Ford was president. There was a scare. Fort Dix soldiers showed up with an H1N1 variant. They were very worried it could be something like 1918. They didn't do a very good job of producing the vaccine, and there was a variety of political things that happened, but basically that virus didn't spread, and we didn't see that again. And right now, the spotlight is on another H1N1, this influenza A 2009, and that's the one we're looking at right now. So, Laura, I'll give it to you.
Thank you very much. Okay.
Well, first I'd like to thank Patty Yelovich for organizing this and inviting us to speak. So thank you very much. Briefly, the outline of my talk, the question was, can influenza pandemics be prevented? I'm going to talk a little bit about human and animal health. That's already been discussed previously by Lynn. A timeline of the current outbreak, briefly talk about the U.S. public health infrastructure, and then intervention strategies. So can influenza pandemics be prevented? The short answer is no. They are, as Lynn mentioned, there are zoonotic disease, and the natural host are wild waterfowl, and unless we want to slaughter all the wild waterfowl from the planet, they're a fact of life on Earth, just like hurricanes, tornadoes, and earthquakes. So we need to recognize that and learn how to live with it. I just want to briefly mention that over half of all the pathogens that infect humans are zoonotic, meaning that they come from animals, and 75% of the newly emerging infectious diseases such as HIV/AIDS, West Nile virus, SARS, Nipah virus, they're all zoonotic, they come from animals. And I want to stress to you that human and animal health are linked. So in terms of the timeline of this current outbreak, the epidemic began in a small town called La Gloria in Veracruz, Mexico, sometime around March 15th. La Gloria is a town of 3,000 people and has a major pig farming industry. The first victim was a 5-year-old boy. He survived. There's not been a connection between the pig farm and the outbreak. And I just want to also mention that while this current virus seems to have some genetic components of pigs, the virus has not been found in pigs, and instead, rather, a Canadian farmer, when he was returning from Mexico, actually inadvertently infected a herd of pigs. So here was a case where a human infected the pigs. But having the term swine flu early on during this crisis confused people, confused governments, and some governments were slaughtering hundreds of thousands of pigs in the mistaken belief that this was going to prevent the spread of the disease, and that's not the case at all. It's already in humans. So slaughtering animals has absolutely no impact, nor does the banning of pork products. The virus doesn't spread that way. It's person to person. I wanted to just show you a picture of La Gloria, Veracruz. It's a very pretty village, and it's along the Gulf Coast. I don't have a laser pointer, but it's along the Gulf Coast down in the bottom of the page. I want to briefly mention to you international health regulations, because this is important. After the 2003 SARS outbreak, in which China, during the initial stages of their outbreak, they first denied having a crisis and then downplayed the severity of it, the UN, the World Health Assembly, decided to revise the international health regulations that hadn't been revised since 1969. And at that time, the 1969 regulations required that only 4 diseases be reported, including smallpox, cholera, plague, and yellow fever. The World Health Assembly approved new health regulations. They were approved in 2005. The new regulations have been taken into effect July 2007, 2 years ago. So this is actually the first outbreak in which these new regulations have been in effect. So that's very important. And I just want to say that the new regulations require that any member state that has a disease within its border, including smallpox, polio caused by a wild-type virus, influenza caused by a new subtype, and SARS. In addition, they must report other diseases that constitute a public health emergency. Now, as alluded to earlier about the different stages of a pandemic, because of the ongoing avian flu crisis, the H5N1, we've been at phase 3, where there's some human infections, but there's no sustained human-to-human spread. During this current crisis, they moved it up to first phase 4, where there's limited human-to-human spread, and then moved it on to phase 5, where there is human-to-human spread, but it's still localized within one WHO region, one World Health Organization region. A pandemic is human-to-human spread across different World Health Organization regions. So this just became available from the MMWR, and I wanted to emphasize that when an outbreak begins, there's usually a lot of confusion in the beginning, particularly in terms of reporting and who has the disease. It appears that the first cases began sometime in mid-March, around March 15th. The Mexican government started getting reports of an influenza-like illness in several areas of the country. But it wasn't until April 12th that the General Directorate of Epidemiology of Mexico reported an outbreak of flu to the Pan American Health Organization, also known as PAHO, in accordance with these new international health regulations. So if these new health regulations had not been passed, it's likely that it wouldn't have been reported because it wasn't required. So we got a very early inkling of what's going on, perhaps in some ways too early, because there was a lot of confusion going on as to what was happening. On April 17th, there was an atypical case of pneumonia in Osaka that prompted enhanced surveillance throughout Mexico. And as you can see where this arrow is, suddenly when you start looking more for a disease, you'll start finding a lot more disease, and you can see how the bars just kind of skyrocket as more cases were found. Now, this chart is confusing, and I know it's confusing, and part of the point of this chart is that it's confusing. That's because during initial stages of reporting of a crisis, people just don't have a handle of what the numbers are. So this is from the CDC, including the CDC and World Health Organization websites. The initial cases in the U.S., it was just reported that there were some cases in California, but they didn't really know how many. Subsequently found out that it was 2 children. But I want to highlight that in Mexico, initially it appeared that there were 854 cases with 59 deaths. And that just really kind of spurred a global panic because, I mean, this is a pretty high death rate. And I think that kind of generated all the hype that you started seeing in the media, along with the name swine flu. People just started jumping to a lot of conclusions. But as the investigation, and WHO subsequently sent a team of experts to investigate, and suddenly the number of cases plummeted down to 26. So, the media likes to have a crisis to report, but sometimes it's a little bit irresponsible to jump so early on when there's so much uncertainty, and that causes unnecessary panic. Now, in May, we have a better handle of what's going on because there's better reporting. And the CDC reported that there were 141 cases on May 1st with 1 death. In the world, it's 367 cases. That's including both the U.S. and Mexico. And the number of deaths in Mexico was substantially lower than the 59 that were reported earlier. And as of yesterday, they reported 642 cases here in the U.S., approximately 6 cases here in New Jersey, although I've just been notified that there might be 2 more. And New York has 90 cases, that's the most in the country, 2 deaths in the U.S. and 29 deaths in Mexico, but no other deaths elsewhere. And in other countries, there's now about 22 countries that are affected. So here's a map as of May 5th of where the spread is. And you can see that most of the cases are in North America and also some cases in Europe and a few cases from travelers from Mexico over to Asia. I want to just quickly talk a little bit about the U.S. public health infrastructure. It's important to note that in the U.S. public health is a state and local responsibility. So the states and localities vary dramatically in how they're able to respond to crises. And the CDC serves in primarily a supportive and advisory capacity. They don't take charge. They make recommendations to the states. Now, I did a couple of years ago an intense study of several states' public health infrastructures, including New York and New Jersey. And I want to highlight to you some of the differences between these 2 states that border each other and just how different they are. So, for example, in New Jersey, we have right now, it's changing, 111 health departments across 21 counties. There's 22 local information network communication sites distributed throughout some of these health departments. Funding is local, so that's through local property taxes. In contrast, in New York, you've got 57 county health departments, including the New York City Health Department, and most of their funding is through the state. So they have a different mandate, they're structured differently. And I would argue, I would pose to you that microbes don't recognize political borders, and the more political borders that you have, the more challenging it is to contain an outbreak. So anyway, that's my plug for consolidation of the 2 Princetons. We do have a unified health department here, but we have 2 governments, so who's in charge? I'm asking you, which mayor is in charge? Now, in terms of an epidemic response, several key factors that health officials use, including surveillance, which I've briefly discussed. And physicians really are the eyes and ears of public health, and you really need to have them engaged, so you really want to have an intact and well-functioning medical infrastructure. And they have to report these diseases. Without their cooperation, you're blind. You really don't know what's going on. You need to know the diagnosis of what's causing the illness. Now, in the cases in Mexico, most of the cases that were reported were in hospitalized patients who were very ill. So we had a biased sample in that most of the patients who were confirmed, laboratory confirmed, were very sick, and we thought that it had a higher death rate than it really did because the people who weren't so sick weren't coming to seek medical care. What's the epidemiology? Investigating the outbreak, how did it start? We still don't know how this started. How does it spread? Well, it spreads people to people. What's the incubation period? Typically the flu is around 4 to 7 days, sometimes a little less. And then there's intervention, efforts to break the chain of transmission, including vaccines, medications, social distancing, and other strategies. In terms of influenza symptoms, well, how many here have had influenza? I'm just curious. How many have had the shot? Very good. Once you're exposed to the virus, it takes about 2 to 7 days to develop symptoms. That's called the incubation period. Symptoms appear suddenly and last for 1 to 2 weeks, and you're all familiar with the symptoms of influenza: fever, body aches, headache. You know, you just don't want to get out of bed. Cough, runny nose. Well, how do you diagnose influenza? Well, you get sick, you go to the doctor, and what they do generally is, well, they'll take a nasal swab and swab your nose and send it to the lab. Now, for the current virus, the CDC recommends that you send the specimen to a state health laboratory that can tell you what kind of influenza it is, whether it's A, B, or whatever. But if you want to find out if it's the current H1N1, then you have to send it to the CDC. So that, of course, takes time. Other influenza tests, such as the rapid antigen test, is really not helpful during an epidemic situation. But in most cases, during your typical flu season, you go to the doctor, and the doctor has seen 5 cases of influenza that day, and you come in with your symptoms, and they'll say, you have influenza, here's your prescription, and go home. That's often what happens. The epidemiology influenza, well, as we heard earlier, it's highly contagious. It's primarily spread through droplet. Of note, it can exist, the virus can exist on non-porous surfaces for up to 2 days. So you can actually touch a contaminated surface, and then you touch your eye, and you're infected. Children are particularly good at spreading the virus because they have higher concentrations of the virus in them, and they can shed it, the virus, for longer periods of time. Now, Lynn had briefly mentioned the R0, or the attack rate. I'm just going to say that, just give you an example, if 5 of you had influenza right now, if the R0 was greater than 1, then you would probably infect the 2 people next to you through coughing and sneezing and things like that. And those people would then spread it on to more people, and it would increase rapidly. If it's only 1, if the R0 is 1, then you will at most infect 1 person next to you, and it wouldn't increase, it would just stay steady. And if the R0's less than 1, then likely you would not infect the person next to you, and the epidemic curve decreases. So what are some of the intervention strategies for influenza? Well, as I mentioned, people can spread influenza without knowing it. They can become infected without knowing it, so that's why containing it is so challenging. And children are highly efficient. So once it gets into a community or a household without an effective vaccine, it's very difficult to contain. I just also want to briefly mention the 1918 pandemic in the Princeton's experience. Princeton at that time, this was during World War I, Princeton was a military camp, and they were able to impose very strict measures here. They basically didn't let anybody off campus. That was a pretty stringent, pretty draconian measure, but it worked. Not one Princeton student died. Now, in contrast, in the town, there were over 300 cases of influenza, 50 cases of pneumonia, and in total 32 people died in the town. And I just want to say I got this picture from the Princeton Alumni Weekly. Published an article on December 17th, 2008, very good article describing what Princeton did during that time. If anybody's interested, I recommend you reading it. Social distancing measures. Now, if it's a deadly epidemic, then you want to close schools and daycare facilities early because, as I said, children are particularly good at spreading the virus. But you need to decide what the trigger is going to be. And you want to keep kids home, which isn't so easy for parents who are working. Kids get stir-crazy. They want to have play dates. It's very difficult. Parents who can work at home, that's optimal, but again, it's not so easy. And people who feel ill should stay home. In other words, everybody needs to work together. Personal hygiene, I'm sure everybody here has been lectured to about having to wash your hands using alcohol gels, avoid shaking hands. Face masks are more controversial because some of the masks might not be effective in filtering out the particles. Only the N95 masks work, and these masks are not made in children's sizes. So the take-home message in all of this is that pandemic influenza is a zoonotic disease. It's a part of life on Earth. We need to live with that. We need to recognize it and deal with it. It's highly contagious. It can be lethal. And your reporting is only as good as your country's medical and public health infrastructures. So if you don't have a very effective, if you have a lot of uninsured patients who don't seek medical care, you might not be catching all your cases, or they might be coming to the doctor too late. And this is reportedly what's also been happening in Mexico. You want strict social distancing measures. Personal hygiene is important, and the communities need to work together. And I just want to also finally give a little plug. My colleagues, Bruce Kaplan, Tom Monath, Jack Woodall, and I have a website called onehealthinitiative.com where we have, it serves as a repository of all things, a repository of information for increasing the communication and collaboration between those who work in animal health and human health, because really that's ultimately what's going to take the measures to deal with influenza. And thank you very much.
Thank you, Laura. So just to get us back to starting a discussion, the issue here is, should we be concerned about this H1N1? And what is our previous experience? It really is important to remember that in 1915, 1916, and 1917, there were increasing death rates from seasonal flu that nobody basically paid any attention to it until we got hit with the 1918-1919 that killed approximately 50 million people. So on one hand, people are concerned, did we hype it on this H1N1? And on the other hand, should we be responding in an aggressive way because of our previous experience? And I will fall on the side of we should really look in with all seriousness at an appearance of a new virus that has not been in human population, that is within 1 month is infecting people in 22 countries in the world. Granted that the death rate is only focused in one country, at least at this point, with 2 death rates, with 2 deaths reported in this country, but this is not simply a call for complacency. As a matter of fact, it has to be taken as seriously as it is humanly possible. Just to finish our presentations, it is really important to realize that we will detect flu, we will detect influenza only if we have a decent global surveillance system. And in some ways, the global surveillance system for influenza has been reasonably good, because that's how we detect early in the year, January through March, what possible new viruses are going to come, and we put them in the vaccine that all of us hopefully will be taking in the September to December or January the following year. So the insistence on supporting the global surveillance is an important element. The unity of animal and public health is an absolute necessity, and this division that here is veterinary medicine and here is human medicine, as Laura and Lynn emphasized, is an artificial division, and we just have to recognize that this is one world in a sense. Even with the diagnostic facilities that we have up till now, we don't really have a good, rapid, inexpensive diagnostic method to have it provided to everywhere in the world. One of the agonies of our country and the rest of the world is we don't pay enough attention to seasonal flu. So our practice of taking the vaccine every year has been modest. And you know, there are people in this audience who know how modest it is, even in the healthcare professionals. So we just get relaxed about flu, and that's very, very important dilemma that we all face. There has to be enough antiviral drugs that are stockpiled in such a way that it can be deployed. The fact that all what the WHO has is 50 million doses of Tamiflu is absolutely a joke. I mean, it's good that we have some stockpile, but it is difficult to see 50 million doses will make an impact if we really face a global pandemic. And then it is very, very important to keep researching and trying to understand those viruses and understand what really makes them flip. Now, for our planned response, there has to be the international health regulations, and the detection, whether it is a clinical or laboratory, is important. But for our country, we have to be absolutely vigilant about surveillance. The license of a new vaccine is a necessity, whether we're going to use it or not, but we have to develop and produce and license a new vaccine against H1N1 that will be available. We need to see that there is a way to distribute antivirals and vaccines if they are available. I mean, I don't know if we have a vaccine today, who will get it and how it will be distributed. I don't think we have an answer to these questions. Also, in a sense, do we have the clinical facilities and the hospital facilities to take care if we really face a pandemic? And in a sense, we are part of the world, so we have to maintain our relationship. So with this sort of just trying to summarize our observations, I want to open it up for discussion. But before we go there, following, because we have to leave this auditorium at 6 o'clock, following, there will be a reception in Schultz, which is just behind us here, and all three of us will stay as long as you want to continue the discussion. Now it's open up to you, and I'll distribute the questions to whoever is going to volunteer to answer. Okay, please.
What's the difference between this H1N1 virus and other H1N1 viruses? Number one, how do we know this?
Okay,
Lynn?
Lynn?
How do we know that H1N1 viruses that we're seeing in these 22 countries, we're already there and we only picked it up because we had to be tested?
So there's several parts to your question. The first thing is remember that there's 11 proteins that this virus makes. H and N are only 2 of them. They happen to be the major proteins that we make antibodies to. And so you can say with some certainty that this virus's H and its N protein are the same because it's a very defined structure that interacts with specific antibodies. So you can say that with some certainty. The other segments, on the other hand, you have a much more difficult time deciding where they came from. Right now, I can tell you, this is very interesting. One of the segments came from a pig virus in Asia, and another segment came from a pig virus in another country, and 6 or 7 of the segments come from birds, and we don't know where they came from. And so each virus, even though there's a constant H and N phenotype that you can assign to them, the genetic makeup of the whole virus is going to be different. And so when you go now with modern technology, and I don't know the answer in detail to this because I've only looked at a few sequences, when the H1N1s are being diagnosed now and they have been sequenced, so we know their whole genome, you can say with essentially very good certainty that they're the same virus. There's a couple of key places people like to look because they're markers of virulence, the hot viruses have them, and so there are ways that you can keep track of this. But as you go back further and further in time, for example, we have essentially re-isolated the 1918 flu virus and have sequenced it, so we can make some statements about that virus in comparison to new ones. The point that I was trying to make is that every virus that comes out is different from the ones that went before because they've been evolving in those different species. So they'll have subtle and not so subtle variations, and so it's a long-winded answer to your question. All right.
Thank you.
Lynn, I didn't get the answer to this question.
Which one?
How many have been sequenced, and how do we know that
Yeah.
they're the ones around the world?
Yeah, that actually, David, I tried to check this morning, and I don't know how many. Our friend, it's a few. It's a few. And one of the things that's really actually encouraging, there is a small open reading frame that's carried in one of the segments that in lethal strains like the 1918 flu had a particular set of mutations in it. This virus doesn't have that set in it. So that much we know, but the numbers are still very low.
So we know for sure that it's not the 1918 virus.
The ones that were sequenced, it's not the 1918 virus. Yeah.
Kai?
In terms of those genetic changes we're talking about, what surveillance measures are in place right now or should be in place to watch for the emergence of more virulent strains?
Laura, do you want to respond to that?
Well, I'm a big proponent of surveillance of animals. So by the time it gets to humans, it's late. And the avian influenza is actually in some ways a success story, because actually when it first emerged in a human in 1997, scientists had been tracking it for a decade before, so they were on top of it. So I would argue that strategies like that, where you're doing surveillance in the wildlife or in domestic farm animals, keeping tabs on what's going on, would be an effective strategy. Of course, you're going to do surveillance of humans, but again, it's going to jump to a human from an animal. Not that I'm aware of, no. The USDA has the Animal Plant Health Inspection Service, and that primarily does inspection of livestock for enforcement purposes to meet health regulations for humans. So there is no CDC for animals. There's no morbidity, mortality weekly report for animals, not for pets, not for livestock, not for wildlife. And perhaps there should be, but again, who's going to do it? Because the Department of Agriculture, their mission is to support agriculture. Their mission is not to do disease surveillance of animals. There really is no one agency that does surveillance of all animals. There's, the Department of Interior will do some oversight of wildlife, but nobody owns the wildlife, and there's not a great lot of support or funding to do that kind of work. So we need to start rethinking some of our priorities in terms of doing surveillance of animals.
Lynn,
just in answer to your question about virulence, there's a very big difference between surveillance and telling, what H and N types you have. That we're reasonably okay with. It gets kind of complicated because to get the various types, sometimes only the CDC has the antibody antigen controls that can tell you what's going on. You can't do some of those in the physician's office. But basic research is telling us that there are certain virulence markers that you can look at it and you can see these. This is why we have to keep going. If we could make a diagnostic kit, a dipstick that turned red when you had that particular sequence or something like that, that would make all the difference in the world. But right now that is just impossible. Almost for any viral disease, diagnosing what you've got, because they go so fast, by the time you figure out what you have, it's too late already. And so this is really important research, this diagnosis, early diagnosis, and specificity. And then in the background of all of that is false positives and false negatives and all the things that drive everybody crazy when it comes to this. And so that is, in your question, there's a very deep set of problems that have to be solved.
Yes, please. Audrey.
So Adel, you mentioned that there's a need for a decent global surveillance system, and Laura, you also mentioned that reporting is only as good as the country's medical and public infrastructure. But one of the limitations of the new IHR is that it doesn't really provide funding for the building of surveillance and the infrastructure. So is this going to spur an increase, what does this mean? Is there going to be an increase will to help provide the funding to certain countries? What does that mean?
It's all based on political will, convincing elected officials that this is something that's important, that needs support. And I think a lot of it should come from grassroots demands to support it. Unless people demand that this is important, elected officials aren't going to make it a priority. But you're absolutely right. The international health regulations do mandate that, reporting be timely. But if you don't have the infrastructure in place, you can't do it. You simply cannot do it. And that's a serious problem for many countries. So you're absolutely right. And again, the elected officials or the leaders of those countries need to be convinced that it should be a priority.
Just to add a little bit to that already. We have one reasonable global surveillance system for influenza. And that is one that is supported globally by funds from both NIH and the World Health Organization. Now, the new international health regulations dictate that each country takes care of itself, because by necessity, WHO doesn't have funds to distribute to develop a system. So consequently, to imagine how the international health regulations are going to be implemented is still a tall order. It was unbelievable. After 40 years, we got the 190 countries in the world to agree on a proposal. They approved it in 2005, as Laura said. It came into effect in 2007, but I doubt that 90% of the countries have an infrastructure inside their own. I mean, the good thing about Mexico here, and one has to recognize that, the good thing about Mexico is they observed increased illness and mortality at the tail end of the seasonal flu season and managed to diagnose it. I mean, one has to be admiring that accomplishment. Remember, SARS was going on in China for 6 months. Nobody heard about it. So whether that is the impact of the international health regulations, the impact that Mexico is across the border, we hear a lot easier about it, I don't know. But in effect, it's a positive step of detecting a syndrome out of season. Yes. Okay, I thought somebody up there. Yes, please.
Pandemics sometimes occur in waves and sometimes return in a more virulent form. Any thoughts on that with this current virus? And if you were the czar of the pandemic, what steps would you take?
I don't want to be a czar. The reason I recounted what happened in 1915, 16, and 17 is exactly your question, which is a very, very relevant question. So the community at large, society at large, when you ask them, has this story been hyped? 37% of our community says, yes, it was hyped. Okay? Now, I don't think the story was hyped in 1916 and 1917 when they were seeing increased mortality and nobody suspected that there is a new virus going on. Now, of course, in 1917, diagnosing a virus infection and getting a Lynn Enquist to look into it didn't exist. But in a sense, one cannot trust what a flu virus is going to do. And consequently, we have to be on our guards. Particularly for this coming season. I mean, it is important. Now we are in May, and the season will start in September. And I mean, I don't want to scare anybody, but this is exactly what happened in 1917. And there were a wave of death from flu in December 1917, including some of the army camps, and basically nobody paid attention to it until it came full-blown in 1918 later on. So your question and your issue here is absolutely important, that we stay with our guards absolutely ready. And that's why I was saying that the issue of stockpiling Tamiflu and Relenza, the issue of, I mean, this is a debate. Should we go on and make a vaccine for H1N1? And I mean, since I'm not the czar, I would say we should. Okay? There is always the experience of 1976 that creates tension. Because remember, there was an H1N1 outbreak of flu in an army camp, actually in New Jersey, and several of the enrollees died. And the NIH and CDC at that time decided to go and make a new swine flu vaccine and started giving it to people, and 26 people or 24 people died. Whether the death was related to the vaccine or not, I don't really know. But it created such a negative impact, and shooting at each other and accusing each other, how dare you guys go and make a vaccine, you didn't know, giving the vaccine, people died from it. If that H1N1 in 1976 would have progressed to be a pandemic and we were caught without a vaccine, we would have been looking sick. And consequently, I think the only diligent answer would be to go on and make a vaccine. Now, that raises another issue, but I don't want to get into, because obviously most of the vaccine capability today is involved in making the seasonal flu vaccine. So you will have to go extra at this point. Yes, Betty.
So next year, when we are getting ready to get our vaccines, the current existing vaccines, is that going to do any good, provide any protection? Should the virus mutate?
So let me just be careful here. The currently existing vaccine is last year's vaccine, has no value. We are making a new vaccine for the new season. I don't know, honestly, its composition. And very unlikely that it will have a cross-protective effect against H1N1. So there is an argument, there is a scientific argument. Should the next seasonal flu vaccine, which is in production now, get added to it an H1N1 component? Or should we make an H1N1 vaccine independent and just the season comes, no H1N1, just give the seasonal vaccine as usual, increase the uptake as much as you can, and keep the H1N1 vaccine just in case of an emergency? I would probably do the latter. Please.
Yes, sir. The period of 1915 to 1918 was over 90 years ago, and the level of knowledge, the status of science, and the existence of institutions was much different than today. And yet we continually make comparisons between the pandemic of that period and what might happen today, when many of the factors that could affect the outcome of a pandemic are very different. Do you have any thoughts, or can you shed any light on that? And what would happen if the pandemic of 1918 were to occur today, given the status of science and the institutions and so forth that exist?
I mean, as Lynn is saying, it could be worse. Do you want to comment first, and then I will follow you?
Well, it's interesting to consider that possibility. I mean, we have better clinical support. We have antiviral drugs. We have, better ways of dealing with patients. But that's only in countries that can handle it. There are many countries out there that are essentially back where we were in 1915, 1916. The other thing is that people move around the world at an incredible rate right now. It is very possible that if that virus ever got going again with the same sort of thing, that it would spread faster, and because we're not really quite prepared, you might have an initial real bolus of some serious things. You can hope that we're going to be smart enough to be able to deal with this. But right now, how many people go to emergency rooms just because they've got the sniffles because of what they heard on CNN or whatever? We're already seeing that we don't have the capacity to handle what's now being called the surge, when all of a sudden a lot of people are coming in. So it's a very important question, but I think you can't assume just because we're smarter and we have better things that it's going to be less well handled.
The amount of traveling that we're doing.
Well, that's
the mobility of people throughout the world.
That's a major thing. Look, yeah. Well, there's more people on the planet now than there were then. But look at where most of the people that are being diagnosed right now came from. They came from Mexico on vacation. They went to Europe. They went to Hong Kong. They went, So it's evidence of the fact that we are a highly interactive group, and there's more of us now than ever. And so we have to be very careful about how we think about this.
David.
They put up an equation.
The R0.
The R0
Yeah.
and all that sort of stuff. The reality is that lots of stuff is the average person knows and needs many more people today than they did in 1950. We live in cities.
Yeah.
We don't live
This is a
on a
mitigating
farm.
factor.
I just want to...
And that gets exponentially.
There are some incredible models now for epidemic spread, epidemiological spread, and one of them was based on dollar bills. So so that's a really fascinating study, because that is probably the best example of human-to-human contact at the distance of about 3 feet. And when you watch the serial numbers spread around, you get an amazing vision. Yeah.
We have in the audience here a senior from ecology and evolutionary biology who modeled flu and its spread in communities. Kai, do you want to comment?
Yes, I guess
I'm setting you up.
we definitely have a very homogeneous mixing population. some of the viruses, the virus that we've seen in Mexico and basically North America in general, jump over other continents. And we've already seen cases in Europe and a few in Asia, and I think one in Colombia. So there's only a matter of time before you see sustained transmission in those continents.
Any other questions? Yes, please.
I have a question about homeopathy, which was used in the 1918 flu epidemic. And I don't recall exact statistics, but I know that a large amount of homeopathy was used, and I think it was less than 1% of the patients that were given homeopathy died, whereas those that were given vaccines and whatever was available at the time did. And homeopathy is known to be very, very effective for the flu virus. What is it talking about? Can't read well.
I don't know what you're talking about, seriously.
Homeopathy, Samuel Hahnemann was a German doctor who founded homeopathy in the late 1800s, and it was used in the United States through the turn of the century. In fact, the only doctor that has a statue in Washington, D.C. is Samuel Hahnemann, in Massachusetts. And there is one in particular called the psilocybin, and I know it's been extremely effective in the homeopathic community for viruses. In fact, a couple of times that I was exposed to it myself when everyone else around me got sick, and I started to get sick, and I took it, and I never got the flu. So I think like there's some discussion.
Well, I mean, unfortunately, I personally am not aware of this compound, but I can categorically state that we have today FDA licensed only 4 drugs against flu. And we have tested them, and 2 of them, the virus, the H1N1 virus, is resistant, and 2 of them are the ones that we are talking about, the Tamiflu and the Relenza. So I'm not sure of anything else that is not FDA approved and how effective it will be. And remember, when things get into society, a lot of additional issues get involved into, and one has to just really stay with what is proven scientifically and licensed for this particular purpose in our country.
Well, actually, it is licensed by the government of the U.S. Homeopathic Pharmacopoeia, and it is licensed with the
I am not aware that it has a proven antiviral effect. I'm sorry to respond that way. It looks like categorical response. I mean, remember, there has been several attempts to say zinc pills will treat flu, this and that. Some of these supplements and some of these ideas are credible enough, but not really rising to the kind of scientific evidence that is important to make it a policy. Yes, please.
Yes. Scientifically, how do the researchers go about determining the doses you get, and whether it might be based on their weight, their body weight, or whatever? But how do they arrive at an estimation of a suitable dose
Well, to have a compound, whatever the compound is, approved for an indication by the FDA, it goes through 3 phases. One phase of it is called dose escalation, whereby you calculate the dose and the effect that you get across several doses. So you come up with the optimal dose. So in the final analysis, when a drug or a vaccine is approved by the FDA, it is approved after those kind of studies determine what is the optimal dose. So that's what we use. There is always room to say, and this is not the effective dose for everybody in society, because we're not really carbon copies of each other. But in general, most phase 3 trials will include thousands of individuals, and any drug is tested in that kind of numbers. So in general, that's the way. One day we will find out that our genetic composition determine our response to drugs, and we might have to get into a completely different story. But I would leave that for the later discussion. Okay, please.
The H1 virus this year was mostly resistant to Tamiflu.
To ranitidine and remdesivir.
Not the swine virus, the virus, the human virus in the community is mostly resistant to Tamiflu. If that virus then is swine or H1N1 were to co-circulate this year, what's the likelihood of the swine virus developing resistance to Tamiflu?
Well, the likelihood of any virus, it doesn't matter whether it's H1N1 or any other type of virus, will one day, sooner or later, develop resistance to the antivirals or antimicrobials that we're using is high. This is the nature of the microbe and the interaction with their environment. The MRSA is a product of bacteria developing resistance to almost all antibiotics that we have. Now, should that deter us from using antivirals if we have an outbreak of flu? The answer is no. But one has to realize that there is a cost for using antivirals against a flu outbreak or any other infectious disease outbreak. And again, we have a thesis here on the possibility of antiviral resistance as you put the pressure on the virus.
Just add one other thing. One can never be sure that just because the virus that's going on right now is Tamiflu resistant, for example, and in its genetic background that's a positive force, maybe only when the drug is around. When the drug's not around, maybe the virus won't be as lethal or as infectious or whatever. And also when it gets into the reassortant mix that's out there, you can't predict exactly what's going to happen. And so, I mean, anytime you treat with an antiviral, as Adel just said, there's a possibility you're going to select for a resistant virus. And if that has a positive selection pressure during the spread things, it'll be carried around. And so we have to watch that. You have to really be careful about what you do. In answer to your question about how you dose, if you don't use enough antiviral drug to stop the thing from replicating or spreading, you're going to select for resistance. But if you use too much, there's always a side effect that's associated with overdosing. So, you know, people say 1 pill is good, 10 must be better. That's a disaster. I mean, you have to, this is a balance. Why the FDA spends so much time doing these escalated dose things is to figure out in a general population, what's the dose that really works. And even when people are 200 pounds or 100 pounds or whatever, are healthy or young or the whole business, it's a very, very interesting and complicated game to play. Yeah.
So, last question.
Thank you. So the last 3 years, the vaccine manufacturers have been focused on H5N1, developing, licensing a vaccine against H5N1. They even went so far as to be able to test and license the pre-pandemic H5N1 vaccine as well. So much that we've had governments take the decision to allocate millions of dollars to the stockpile of these pre-pandemic vaccines. It's H1N1 now that we're dealing with. So now the focus is shifted to this other strain as vaccine manufacturers and of course our agencies rush to figure out how to develop a strain against this. How similar do you think these strains are in their ability to induce the kind of immune response that we need in an effective vaccine? And is that pathway there for us right now to be able to do that within the time, in a shorter amount of time frame that we need to try to help?
I mean, unfortunately, the cross-reactivity between the different viruses is very little at this point. So we probably will need another vaccine for the H1N1. Now, the question and the challenge is back to Professor Enquist. I mean, somebody has to devise for us a way to induce immunity or to produce a vaccine against flu that is not dependent on the variable part of the virus that is quickly changing from one season to another and from one pandemic to another, and come up with a vaccine that is pan-flu vaccine. That has been a dream in some ways. But it is the ultimate solution. And is it that far? I don't think it's that far with the capabilities of molecular biology and genomics and so on that we have today. But until we get to that point, we'll have to depend on making a vaccine for whatever virus is threatening us. Is that waste of money? Is that waste of resources? I think no, because if we get hit seriously with one of these pandemics and we didn't have a vaccine for it, it will be a very, very tough time for all of us to face it. I think at the end, I just want to thank Professor Enquist and Laura Kahn. They have been fantastic. And thank you all for terrific questions. So.